Hello, welcome back to my asynchronous class of Biopsychology 1. My name is Advika and today's topic is about vision. And in this video, we'll be reviewing the first module, which is the visual coding. So we will be talking about the general principle of perception at first, and then the eyes and its connection to the brain, continuing to visual receptor, and finally the color vision. First of all, let's check the principle of perception first. Yeah, the old misconception is that the notion that our eyes send out side rays so we can see. So actually, this principle was probably the first scientific discovery in psychology. However, the Arabic philosopher called Ibn al-Haytham, he kind of like observed when you open your eyes at night, you immediately see the distant stars. And based on that observation, he reasoned that if you saw by sending out side rays, they couldn't get to the stars that fast. Then he demonstrated that light rays actually bounce off an object in all directions, but you see only those rays that reflect off this object and strike your retina. So when you see a tree, for example, your perception is not in the tree, it is actually in your brain. So you see something only when the light from the objects alter your brain activity. Let's say even if you did send out rays from your eyes, which is not, or when they struck some object, you wouldn't know about it unless they bounce back and return to your eyes. So actually, the principles of this perception is actually caused by the brain that codes information largely in terms of which neurons are active and how active they are at any moment. So actually impulses in certain neurons indicate light, whereas impulses in others indicate sound, touch, or other sensation. And in 1838, Johannes Müller described this insight as the law of specific nerve energies. So actually Müller held that whatever excites a particular nerve then establishes a special kind of energy unique to that nerve. So in modern terms, the brain somehow interprets the action potential from the auditory nerve as sounds, those from the olfactory nerves as odors, those from the visual nerve as vision, and so forth. So let's do some demonstrations. Try to rub your eyes, and then you may see some spots or flashes of light even in a totally dark room that you can still see lights. When you did this, you actually applied mechanical pressure, which excited a visual receptor in your eyes. So actually, this is like anything that excites those receptors is perceived as light. And if you decided to do this demonstration, just make sure that you remove any contact lenses and shut your eyelids and rub your eyes gently, all right? Okay, so to get a better understanding about our visual system, let's take a look at our eyes first. As you might remember from your biology class in your high school, that lights enter the eye through an opening in the center of the iris called the pupil. And then it is focused by the lens, which is adjustable, and the cornea, which is not adjustable, and then projected onto the retina. It's the rare surface of the eye, which is lined with the visual receptors, right? All right. So, and then we'll continue to take a look of the visual system of the vertebrates. So let's say if you're designing an eye, you will probably send the receptors messages directly back to the brain, right? However, interestingly, in the vertebrate retina, messages go from the receptors at the back of the eye to bipolar cells that are located closer to the center of the eyes at first. As you can see from this figure, in this boat figure, as you can see the bipolar here, bipolar cells here. And then it continued to send the message from the bipolar cells, send the message to the ganglion cell. And then also located still closer to the center of the eye, and then the ganglion cells, axon, join together and travel back to the brain. And then there is an additional cells called amacrine cells that get information from bipolar cells and then send it to other bipolar, amacrine, and ganglion cells. And these amacrine cells rewind the input to ganglion cells, enabling certain ones to respond mainly to particular shape, direction of movement, changes in light, in color, and other visual features. And researchers actually have identified dozens of types of ganglion and amacrine cells varying in their chemistry and connection. The other things that is important from our visual system is a blind spot. Yeah, so it is actually a consequences of our visual system. So the ganglion cell axons join to form the optic nerve that exits through back of the eyes, right? The point at which is leave, also where the blood vessel enter and leave, yeah, is a blind spot because it has no receptor there. In everyday life, you may never notice your blind spot for two reasons. First, your brain fills the gap and then the second one is anything in the blind spot of one eye is visible to other eye. Okay, so let's continue talking about the fovea and periphery of the retina. So when you look at details, such as when you're looking at the letters on the screen, you actually fixate them of the central portion of your retina, especially the fovea. So the fovea is a tiny area specialized for acute detailed vision. Because of blood vessels and ganglion cell axon are almost absent near the fovea, it has nearly unimpeded vision. And the tight packing of receptors aid perception of details. And then more importantly for perceiving detail, each receptor in the fovea actually connects to a single bipolar cell, which is turned to connects to a single ganglion cell that has an axon to the brain. The ganglion cell in the fovea of humans and other primates are called midget ganglion cell because each is small and respond to just a single cone. That is, each cone in the fovea has a direct route to the brain. Because the midget ganglion cells provide 70% of the input to the brain, remember that, your vision is dominated by what you see in and near the fovea. All right, and then toward the periphery of the retina, more and more receptors converge onto bipolar and ganglion cells, yeah? And as a result, the brain cannot detect the exact location or shape of a peripheral light source. However, this summation enables perception of fainter lights in the periphery. So in short, foveal vision has better acuity or sensitivity to detail, while periphery vision has better sensitivity to dim light. And then the vertebrate retina contains two types of receptors, which are rods and the cones, as you can see in the figure here. So the rods is actually abundant in the periphery of the human retina, and it responds to faint light but are not useful in daylight because bright light bleaches them. And then the cones is abundant and near in the fovea, and they are less active in dim light, and they are most useful in the bright light and essential for the color vision. And because of the distribution of rods and cone, you have good color vision in the fovea, but not in the periphery. All right, still about the rods and the cones. So although rods outnumber cones by about 20 to 1 in the human retina, cones actually provide about 90% of the brain's input. So remember that the midget ganglion cells, it's inside the fovea. Each cone has its own line to the brain. So in the periphery, mostly rods, each receptor shares a line with tens of hundreds of others. And then overall, 120 million rods and 6 million cones converge onto 1 million axons in the optic nerve on average. And then, 1 ratio of rods to cones may sound high, but the ratio is much higher in species that are active at night, such as South American oil birds, which live in the caves and emerge only at night. They have about 15,000 rods per cone. As a further adaptation to detect faint lights, their rods are packed 3 deep throughout the retina. So people also vary substantially in the number of axons in their optic nerve, and the size of the visual cortex. It's largely for a genetic reason. Some people have 2 or 3 times as many axons from the eyes to the brain as others do. And also they have more cells in their visual cortex and greater ability to detect brief, faint, or rapidly changing visual stimuli. And then heightened visual response is also valuable in many activities, especially in sport that require aim. And then also researchers find that the top performers in tennis, squash, fencing, baseball, and badminton, they usually show enhanced processing of visual stimuli compared to other people. Right? Of course, excellent vision is hardly the only ingredient for athletics success, but it is actually help. And then both rods and cones actually contain photopigments. Yeah, it is a chemical that releases energy when struck by the light. And photopigments consist of 11 cis retinal. It's a derivative of vitamin A. It's bound to protein called opsins, which modify the photopigment sensitivity to different wavelengths of light. And here is the summary of the foveal and peripheral vision that you can read in detail by yourself. And continuing to the color vision. So the visible light consists of electromagnetic radiation within the range from less than 400 nanometer to more than 700 nanometer. So we perceive the shortest visible wavelength as violet, as you can see in this picture. And then progressively longer wavelengths are perceived as blue, green, yellow, orange, and red, as you can see in this right side of this figure. So we call this wavelength lights only because of the receptors in our eyes are tuned to detect them. So if we had different receptors, we would define light differently. Indeed, many species of bird, fish, and insect have visual receptors sensitive to what we call ultraviolet radiation. But of course, we cannot know what it looks like to them, but so far as they are concerned, ultraviolet radiation is a type of light. And in some species of bird, the male and female look alike to us, but different to bird, because the male reflects more ultraviolet light. All right, so people actually like distinguish red, green, yellow, blue, orange, pink, purple, greenish blue, and so forth. And presuming that we don't have a separate receptor for every possible color, so how many actually receptor types do what we have? So according to trichromatic theory of color vision, or the Young-Helmholtz theory, we perceive color through the relative rates of response by 3 kinds of cones. Yeah, each one maximally sensitive to a different set of wavelengths. So people could match any color by mixing appropriate amounts of just 3 wavelengths. Therefore, he concluded that 3 kinds of receptors, we now call them cones, are sufficient to account for human color vision. And this figure shows wavelength sensitivity function for the short wavelength, medium wavelength, and long wavelength cones type. So each cone responds to a broad range of wavelength, but to some more than others. So according to this theory, we discriminate among wavelengths by the ratio of activity across the 3 types of cones. So for example, light at 550 nanometer excite the medium wavelength and long wavelength receptors about equally, and the short wavelength receptor almost not at all. And these ratios of responses among the 3 cones determines a perception of yellow-green. So more intense light increases the activity of all three cones without much change in the ratio of responses. And then as a result, the light appears brighter, but still the same color. So when all three types of cone are equally active, we see white or gray. All right, so given the desirability of seeing colors in all locations, we might suppose that three kinds of cones would be equally abundant and evenly distributed in our nervous system. But in fact, there are not. Long and medium wavelength cones are far more abundant in than short wavelength or the blue cones. Consequently, it is easier to see tiny red, yellow, or green dots than the blue dots. So you may try this. You can look at the dots in the following display. You probably, first you may see it from close, and then from after that from greater distance. You probably will notice that the blue dots look blue when you see it from close distance, but appear black from a greater distance. And the other colors remain distinct when the blue is not. All right, so actually the tree chromatic theory is incomplete as a theory of color vision because, for example, because of this demonstration, you can actually try this. Pick a point near the center of the figure and stare at it under bright of light without moving your eyes for a minute. And then you can look at the plain white surface after you see the picture. Plain white surface, maybe like a wall or blanket sheet of paper, and try to keep your eyes steady. What happened? You will see a negative color after image, a replacement of the red you had been staring at with green, and green with red, yellow and blue with each other, and black and white with each other. So actually, to explain this and related phenomena, Ewald Hering, he's a 19th century physiologist, proposed the opponent process theory, that he proposed that we perceive color in terms of the opposites. That is, the brain has a mechanism that perceives color on a continuum from red to green, another from yellow to blue, and another from white to black. So after you stare at one color in one location long enough, you fatigue that response and then swing to the opposite. Part of the explanation for this process pertains the connection within the retina, actually. So for example, imagine a bipolar cells that receive excitation from a short wavelength cone and inhibition from long and medium wavelength cones. So actually it increases its activity in response to short wavelength or blue, and decreases in a response to yellowish light. After prolonged exposure to the blue light, the fatigue cell decreases its response. And because of a low level of response by that cell usually means yellow, you perceive yellow. So when researchers around 1950 first demonstrated that certain neurons in the visual system increase their activity in response to one wavelength of light and decrease it to another, they revolutionized our understanding of vision and of the nervous system in general. So, however, the explanation about the opponent process theory, it cannot be the whole story. So you may try this experiment. So you might stare at the X in the following diagram for about a minute under a bright light. Yeah, make sure that you see it under a bright light, and then look at a white page. Okay. For the after image of the surrounding area, you might saw red color, as the theory predicts. But what about the circle inside? Theoretically, you should see a gray or black after image. It's the opposite of white, right? But in fact, if you used bright enough light, you saw a green after image. What you saw in the surround influenced by what you saw in the center. And here's another demonstration. You may first look at the figure And then note that although it shows 4 red quarter circle, you have the illusion of a whole red square, right? Now stare at the X in the figure for at least a minute under the bright lights, and then after that, look at a white surface and see what happened. People usually report that the after image fluctuates. Sometimes they see from 4 green quarter circles, and sometimes they see a whole green square. So if you see a whole green square, it is the after image of an illusion, and the red square you saw wasn't really there. This demonstration suggests that after image depends on the whole context, not just the light on individual receptors. The cerebral cortex must be responsible, not the bipolar or ganglion side or the ganglion cells. All right, so let's continue. The trichromatic theory and the opponent process theory cannot easily explain color constancy or the ability to recognize colors despite changes in lighting. So let's say if you wear green tinted glasses or replace your white light bulb with a green one, you still can identify bananas as yellow, paper as white, and so forth. So your brain actually compares the color of one object with the color of another, in effect subtracting certain amounts of green from each. To illustrate, examine this figure. Although different colors of light illuminate the two objects at the top, you easily identify the square as red, yellow, blue, and so forth. And then note the result of removing context. Yeah, the others show that the square that looked blue in the top left part and the yellow, yeah, without the context that indicated yellow light or blue light, all these squares looks gray. For this reason, we should avoid talking about the color of a wavelength of light. A certain wavelength of light can appear as different colors depending on the background. And to account for colors and brightness constancy, Edwin Land, he's the scientist, he's proposed the Retinex theory. This is a combination of the words retina and cortex. So the cortex compares information from various parts of the retina to determine the brightness and color for each area. And continuing that, another researcher, Dale Pearce, and the college have expressed a similar idea in more general terms. So whenever you see anything, we make an inference. For example, when you look at the object in the figure, you ask yourself, on occasion when I have seen something that looked like this, what was it really? So you go through the same process for perceiving shapes, motion, or anything else. That is, visual perception requires reasoning and inference, not just retinal stimulation. Similarly, we perceive the brightness of an object by comparing it to other objects, right? So examine this figure. So the object in the center appears to have a dark gray top and white bottom, right? Now cover the border between the top and the bottom with your finger. You see that the top of the object was exactly the same brightness as the bottom. All right, so let's moving on to the color vision deficiency. So one of the first discovery in psychology was color blindness, actually. So it better described as color vision deficiency right now. So complete color blindness or perception of only black and white is actually very rare. And then the color deficiency is result because of people with certain genes fail to develop one type of cone or develop an abnormal type of cone. So in red-green color deficiency, the most common form of color deficiency, people have trouble distinguishing red from green because their long and medium wavelength cones have the same photopigment instead of different one. So the gene causing this deficiency is on the X chromosome, yeah? So about 8% of Northern European men and a smaller percentage of men from other backgrounds are red-green colorblind compared with less than 1% of women. So actually women with one normal gene and one color deficient gene, and that includes all women with green color deficient father, are slightly less sensitive to red and green than the average for other people. All right, so that's all for visual coding topic. I'll see you shortly in the next video about vision. Thank you for your attention and bye.